So the next topic that we are going to discuss under chapter 7, which is nucleic acids and proteins, we are going to discuss about translation. We have previously discussed this topic in the chemistry of life, but that is only for, it's basically a foundation topic wherein both SL and HL students are supposed to learn this process. But now, the chapter 7, okay, 7 onwards, these are the topics that are supposed to be learned by HL students. So we have to give more depth in the details that we are supposed to teach about these topics. So, well, first things first, students should have a knowledge regarding the RNA types, because we have previously discussed that we have different RNA types such as mRNA, tRNA, ribosomal RNA. So by knowing the differences between the three structures, then the students would be able to know when to use it in the process of protein synthesis. The process of translation is the second process that is involved in the protein synthesis, wherein this is the point where we are actually building the polypeptide chain that is supposed to be built into a protein. Okay, so before the translation process, okay, so the students should already know that the process of translation is occurring in the 5' to 3' direction. Okay, so it is applicable in the process of replication and transcription as well. And prior to the translation process itself, the student should be able to know the process of the tRNA and the tRNA amino acid activation. Okay, so in the case of tRNA amino acid activation, the student should be able to learn that in each and every, since there are 20 amino acids, there is a specific tRNA that is supposed to be used for each amino acid. So we are only able to bind a tRNA with a specific amino acid using a specific amino acid tRNA enzyme. Okay, so each amino acid is linked to a specific tRNA by the action of the tRNA activating enzyme before it is used in the process of protein synthesis. Okay, so first, the student should be able to know or should have a knowledge of the tRNA structure. So in the case of the tRNA structure, the students should be able to know that they should know that the structure is also single-stranded, but it is, this time the tRNA is actually folded, or it's actually formed into a clover-shaped structure, wherein at the other end, okay, so for example, if we have a tRNA structure here, okay, so for example, this is our tRNA structure. At one end of the 3' end of the tRNA, this is where we are going to attach our amino acid. Okay, with the help of the tRNA activating enzyme. And the other end of the tRNA structure, this is actually the anticodon. It is also a triplet code that is only complementary to the triplet code of the mRNA, which is the codon. Okay, so we're able to use the tRNA by reading through the triplet codes present in the mRNA, so that it will be able to condense, or it is able to put together or build the amino acids together into a polypeptide chain. Okay, so right after discussing the tRNA structure, okay, so tRNA is only able to attach onto the codon, okay, of the mRNA through complementary base pairing. So that by now the student should already learn what is complementary base pairing. Okay, so right after, okay, so each amino acid Okay, so we are only able to explain this process wherein, for example, if we have our tRNA structure here, okay, so we are only able to attach the tRNA with a specific amino acid, okay, with the help of using energy in the form of ATP and a specific enzyme that is only working with a specific amino acid. So, amino acid enzyme, sorry, tRNA activating enzyme. Okay. So with the help of the ATP and the tRNA activating enzyme, we're able to attach the amino acid with the specific tRNA. So therefore, in the end, we are able to use this tRNA in the process of translation. Okay, so let's just say this is going to be the amino acid. Okay, so the next part, okay, the next thing that is the student is needed to learn is the ribosome structure. Although this is not discussed in the core part, the HO student should be able to know the difference between the three types of the RNA. This time, ribosome is actually another type of RNA. Actually, it is a combination of a protein and the ribosomal RNA. So in the case of ribosome structure, okay, it is made up of two subunits, namely the large subunit and the small subunit. Okay, so in the small subunit, this is where we can find the mRNA binding site, where the mRNA binds itself before it is going to be used in the process of translation. And in the large subunit, this is where we are going to find our tRNA binding sites. And by tRNA binding sites, there are three tRNA binding sites. Okay. So if we are able to provide a diagram for the structure of the ribosome, so much the better. So okay, let's say this is the large subunit and then the small subunit. So the space in between the large and the small subunit is where we are going to attach our mRNA, so that it's going to be read by the ribosome later on. Okay. So within the large subunit, we can find three binding sites, namely the exit site or the E site, the P site or the peptidyl site, and the A site. Okay, so you have to make sure that the students should learn that the sites that is going to be used accordingly, that is going to be used chronologically, is the P site. The P site is going to be used first, and then the A site, and then the E site. Okay, so the first tRNA is going to attach onto the P site, and then the incoming tRNA is going to attach onto the A site. Okay, so later on I will be discussing the further process of translation, which includes initiation, elongation, translocation, and termination. Okay, so it has been noted already that the process of translation, before we are able to explain the process of translation, we have to reiterate to the students that they should know the difference between the three tRNA binding sites. Within the large subunit of the ribosome, there are three binding sites, namely the E site, the P site, and the A site. Okay, so the P site is going to be the first binding site of the tRNA, where the first tRNA is going to attach. Okay, and then the A site is where the second tRNA is going to attach itself. Okay, by complementary base pairing based on the triplet codes or the codons that is present on the mRNA. And then the E site here, okay, this is where the tRNA leaves the ribosome. When the tRNA is already empty and it has already left its amino acid with the polypeptide chain, then the free tRNA can now leave the ribosome through the exit site. Okay, so now we can now explain the process of initiation. The process of initiation starts when the ribosome recognizes the start codon. The start codon represents the triplet codes of A, U, G. Okay, so we have to make sure as well that the nitrogenous base U is only present in RNA. That's why we have the AUG here. Okay, so when the ribosome has recognized the start codon, which is AUG, then the appropriate tRNA binds on the P site. This time, the appropriate tRNA here is UAC. It's because the binding of the tRNA with the codon works by complementary base pairing. So basically, the codon of the mRNA is a complementary triplet code of the anticodon of the tRNA. So this time, if our start codon here is AUG in mRNA, the triplet code or the anticodon in the tRNA is UAC. Okay, works by complementary base pairing. So that is initiation. So now the next tRNA or the incoming tRNA is going to attach on the A site, okay, depending on the triplet code that is going to be present here in the mRNA. Okay, so the next process that is going to be discussed is the process of elongation. Okay, so in the process of initiation, therefore we are able to attach the tRNA with the amino acid that is attached to it. So when two tRNAs that are attached onto the P site and A site are attached or binded on the tRNA binding sites together, automatically the amino acids that is attached on the tRNAs, there would be a peptide bond that is going to be built or is going to be formed in between the two amino acids. Okay, so in the process of elongation, we are able to elongate or to build our polypeptide chain through the process of elongation when more and more incoming tRNAs are going to attach onto the A site.